It's my great pleasure to share our work. My title is Cellular Label-Free Integrative Pharmacology Assays. Identify active compounds in traditional Chinese medicine. The content included background principle, application, identification of active compounds, and conclusion. TCMs have reliable clinical efficacy, which are important source for drugs. UU2 discovery artemisinin to treat malaria, which won Nobel Prize in Physiology or Medicine. In this period, 1,881 drugs were approved, and 15% are related to natural products. Therefore, natural products are important source of drugs. However, TCMs are complex. It contains hundreds and thousands of compounds. They vary content. This is the components. It's very complex. So there are two key questions. What are the active ingredients? How do they work? To solve this problem, our group leader proposed the problem. It aims to comprehensively decipher the composition, structure, and function of TCM, construct simple resource library, elucidate the integrative mechanism of TCM, and lay a foundation for interpreting the scientific nature of TCMs and developing new drugs. This is the research protocol. Prescription was considered as one object, and its effective herb can be obtained. These herbs were separated into multi-component fractions and compounds. This is the separation and characterized process. It clarifies the component of TCM. These fractions and compounds were screened by clip assay to find active compounds and fractions. These active compounds and fractions were evaluated by ADMET system, biological, and clinic to obtain novel TCM. This is the functional optimization and biological evaluation process. It clarifies the TCM mechanisms. There are three key techniques for hemoglobin projects, including high-quality sample library, high throughput and high content screening, and separation capability. Currently, pharmacological assays included molecular assay and phenotypic assay. This assay use artificial system and are biased to a special mechanism of action. It is high throughput but poor correlation. This assay examines drug active in inactive cell, T cells, or animals. It has good correlation but low throughput. CLIP as new generation technique. It converts drug-induced dynamic redistribution of cellular constituent in cells into an integrated and kinetic response, termed dynamic mass redistribution. It has advantage of high throughput, real-time recording, wide target pathway coverage. It has been used to study GPCR ion channel tyrosine kinase. These characters makes it a suitable approach to study target pathway of TCMs. GPCR are the most intensive studied drug target. Now, about 34% of all drugs approved by FDA act at 108 GPCRs. So it is important to study this kind GP receptor. This receptor grouped into GQ, GS, and GI subtype. Upon ligand bonding, each class of DMR singles show different kinetic and dynamic characteristics. So CLIP assay can reflect the unique single pass of GPCRs. Until now, we have built 19 kinds of GPCR target model, including adrenergic receptor related to asthma, opioid receptor related to pain, dopamine receptor related to Parkinson's disease, muscarinic receptor related to asthma, and free fatty acid receptor related to diabetes. And often GPR certified related to inflammation. This is an example of target pathway deconvolution of neurotensin receptor. This receptor play important role in cancer, pain, and obesity. We use different inhibitor to probe the pathway. We also characterized FFA4. We evaluated 4 classes and obtained the target pathway. Given that TCM is complex, so we use CLIP combined with 2-dimensional liquid chromatography. This is an example. Curcuma has an important application in treating asthma, and active beta-2 receptor can dilate bronchi. So we suspect maybe in this herb contain beta-2 agonist. We perform the first dimensional prepare and CLIP screening and found these 2 fractions are active. We then performed the second dimensional prepare and found these 2 active fractions are difficult to separate it on the same column, while it can be well separated on X-chart column. We screening this fraction and found 6 active fractions. Finally, we prepared 3 compounds and found this one is highest potency. This is the first time to discover this type compounds could active beta-2 receptor. We also proposed the multi-technique integration. We perform HPLC preparation in analysis level and quantify it online for CLIP screening. We use LC mass to predict active compounds for aim-oriented preparation. This method has advantage of the analytical and biological screening were on the same scale. The CAD quantification ensures accurate screening on micro scale. The composition of mass identified accelerates the process in identifying active compounds. We use the method to identify FFA4 agonist. We prepared 10 fraction. We compared the porosity of CAD and UV and found for compounds with different structure, CAD is more precise than UV. We screening these 10 fractions and identify their structures and found zirconia show the best potency. Then we study the pathway of zirconia. Zirconia could trigger dose dependently response, and the response signal could be absolutely blocked by antagonist. We verify the activity by Blipper and test its receptor specificity. Also use H inhibitor to prove the pathway. Finally, we evaluate its diabetic effect and found it could reduce plasma glucose level compared to control. Also, separation material is critical. Until now, we have developed over 100 types of separation material to meet TCM's separation. Based on theoretical model, we proposed column selective spider. This can provide column system for separation TCM. Now, for separation pool, mid-pool, non-polar compounds, different systems were designed. Now we have built a high-quality sample library for find more active compounds. Conclusion: CLIP as the new generation phenotypic assay. It has a great potency in discovering active compounds in TCM, as well as elucidate their mechanism of action. Due to the complexity of TCM, CLIP combined with advanced separation and preparation methods offer an effective means for discovery of active compounds in TCM. In the future, 3-point aspects should be strengthened, including data analysis method, new biosensor technique, and effective purified method for obtaining active compounds at the low content level in TCM. Thank you very much for your attention. Welcome to Dalian. Welcome to DICP. Thank you.